Hello everyone, my name is Dr. Jamie Locke and I am a transplant surgeon at the University of Alabama at Birmingham Hersink School of Medicine, and I'm really pleased today to represent our entire UAB xenotransplantation program that really consists of individuals across our institution, from nephrology to surgery to pathology to epidemiology to our folks in our human immunogenetics laboratory. And it's really a privilege to represent this work. You know, I think many people may or may not be familiar with the fact that there are 37 million Americans who carry the diagnosis of chronic kidney disease. Almost 800,000 of those individuals also carry the diagnosis of kidney failure and are in need of a life-saving kidney transplant. Kidney transplantation has been shown to be the gold standard. It's in fact the cure for end-stage kidney disease, and it is consistent with a survival benefit over remaining on dialysis. The problem, why can't we give a kidney transplant to everyone in need, is this really unmitigated organ shortage crisis. So I told you there are 37 million people with chronic kidney disease, 800,000 with kidney failure. We only waitlist about 80 to 100,000 individuals any given year for a kidney transplant, and we perform fewer than 26,000 every year. So you can see the gap between supply and demand is vast. It's almost unimaginable how we might actually overcome this. And of course, we need to overcome it. I just told you we have a cure for the disease, and yet we don't have equity in access, and how do we do that? And we believe here at UAB, along with many others, that we have to tackle this by even looking beyond human organ sources for donations, but actually think about how we might change things, change the paradigm, and look for an alternative organ source or organ supply. And this is really where the field of xenotransplantation comes into play, where you can genetically edit a pig in this case and have that pig's kidney be human enough that the human immune system can tolerate it just like it would in human-to-human transplantation. But to do this, you still have to have immunosuppression. And one of the things that we have sought to do in this study is really identify the optimal immunosuppression regimen for pig-to-human xenotransplantation, one that's already FDA approved with standard medications that we as providers are very comfortable with. And that's something that's been quite challenging to do using a non-human primate model, in which many of the medications that we have to use are not in fact FDA approved and in fact remain experimental. Thus, we leverage the Parsons model, which is a model of human brain death, and we perform pig-to-human xenokidney transplants in this model. And I'm really excited to introduce and highlight one of our trainees, Dr. Maggie Jones-Carr, who's going to explain to you our study and our findings.
I'd like to highlight the methods and discoveries of our study. Studying brain-dead human decedents, now known as the Parsons model, was named after our first decedent. We were able to study the human immunologic response to porcine kidney xenografts. We studied a total of 3 decedents. The first received an immunosuppressive regimen without complement inhibition, and the immunosuppressive regimen was otherwise identical for the decedents. This included methylprednisolone, antithymocyte globulin, rituximab, tacrolimus, mycophenolate mofetil, and prednisone, all of which were FDA-approved medications. After xenotransplantation of the first decedent, the xenografts showed evidence of membrane attack complex staining, or MAC, and subsequent thrombotic microangiopathy by postoperative day 1 as well as postoperative day 3. This indicated that the innate immune system was fighting the porcine xenografts with complement, basically poking holes in the cell membranes with this membrane attack complex, which can lead to graft failure. The second and third decedents, their immunosuppressive regimen included eculizumab, an FDA-approved complement inhibitor, 24 hours before and 24 hours after xenotransplantation. This medication is commonly given to kidney transplant recipients with atypical hemolytic uremic syndrome, so it is well studied in this population. The second decedent was maintained for 3 days postoperatively, and these xenografts never showed any signs of MAC deposition or thrombotic microangiopathy, indicating successful immunosuppression with the eculizumab. The third decedent was maintained out to 7 days postoperatively. While this decedent's xenografts never had thrombotic microangiopathy, they did have MAC deposition at postoperative days 5 and 7, which was concurrent with subtherapeutic eculizumab levels. This indicates to us that eculizumab will have to be redosed, just as it is with atypical hemolytic uremic syndrome. In conclusion, pig to human kidney xenotransplantation seems to be immunologically feasible under the cover of complement inhibition at C5 and standard FDA-approved immunosuppression. Importantly, all of these medications have regulatory approval and established safety in humans. Our study, while it was a short-term case series, did provide important insights and will help pave the way for future clinical trials.